Next, we have artificial asexual reproduction, also referred to as artificial propagation. Humans use these methods to produce large numbers of plants that are genetically identical to the parent. This allows for faster multiplication of desirable plants and ensures uniformity in traits such as flower color, fruit quality, or leaf shape. One common method of artificial propagation is cuttings. In this technique, a shoot is cut from the parent plant and placed in moist soil or water. The lower part of the shoot develops roots, while the upper part continues to grow into a new plant. This process is known as vegetative propagation by cuttings. The newly formed roots absorb water and minerals, supporting the growth of the plant. To improve the success rate, rooting hormones are sometimes applied, which stimulate root formation and help the cutting establish more quickly. As shown in the image, the plant is prepared for propagation by cuttings. A shoot is taken from the parent plant, with the lower leaves and flower stems removed. The cutting is then placed in water or moist soil, making sure the nodes are covered to allow root development. Over time, the cutting forms roots and grows into a new plant that is genetically identical to the parent. This method is used by humans to produce large numbers of identical plants. Another method of artificial asexual reproduction is tissue culture. In this technique, small pieces of plant tissue or even individual cells are encouraged to divide under sterile laboratory conditions. These cells can grow independently into complete plants. Often, the tissue first forms a callus, which, when treated with appropriate growth substances, develops roots and shoots. Plants produced through tissue culture are genetically identical clones of the parent and are typically free from diseases, making this a highly effective method for mass propagation. This diagram illustrates the process of tissue culture. Small pieces of plant tissue or individual cells are placed on a nutrient medium in a sterile laboratory environment to encourage cell division. The tissue may form an undifferentiated mass called the callus. When treated with specific growth hormones, the callus develops roots and shoots, eventually forming a tiny plantlet. The plants produced are clones of the parent and are usually disease-free. This method enables the rapid production of large numbers of identical plants.